In the last class, we have discussed about the parts of female reproductive system. Today, we are going to discuss about the process of spermatogenesis and the structure of sperm. Before going into the process of spermatogenesis, you should be familiar with the gametogenesis. So, I'll explain about the gametogenesis. What is meant by gametogenesis? It is the process of formation of gametes. Which are the gametes? Sperm and ova. So, I will define gametogenesis in another way. Gametogenesis is the process of production of ova or sperm from a diploid cell through the meiotic division. I will repeat once more. Gametogenesis is the process of production of ova or sperm from a diploid cell through the meiotic division. Now, you may have a doubt about meiotic division, isn't it? What is meant by meiotic division? It is a kind of division which may happen during the time of gametogenesis. Actually, our body have two types of division. That means our body cells will undergo two types of division. One is mitotic division and the other one is meiotic division. As you know, our body cells are multiplied through division, isn't it? In our body, two types of divisions are there, mitotic division and meiotic division. In mitotic division, a single cell can produce two cells with the same number of chromosomes. That means, if a cell containing 46 chromosomes is dividing, as a result of that, new daughter cells will form. These new daughter cells may also have the same number of chromosomes. That is, the new daughter cells also may have the 46 chromosome in it. Understood? These kind of divisions are known as what? Mitotic division. And in the case of meiotic division, it will happen during the time of gametogenesis. And in meiotic division, a single cell will divide to form 4 haploid cells. And the chromosome number of this haploid cell will be always lower than that of the parent cell. That is, if the parent cell had 2n number of chromosomes, the newly formed cell may have n number of chromosome only. That is, 23 chromosome only will be there in the case of daughter cells. You might have understood the main difference between mitosis and meiosis, isn't it? What is mitosis? In mitosis, the newly formed cells may have the same number of chromosome like that of the previous parent cell. But in the case of meiosis, the newly formed cell may have a half number of chromosome when compared to the parent cell. This is the main difference between the mitosis and the meiosis. Understood, children? I think you might have understood what is meant by mitosis and meiosis. Now, let me show you a video of mitosis and meiosis. It will make the concept more clear. Okay.
Have you ever wondered why children do not look exactly like their parents? Or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells.
As I told you earlier, gametogenesis is the process of production of sperm or ova, isn't it? Now we are going to study about the process of production of sperm or spermatogenesis. So, can you define spermatogenesis? What is spermatogenesis? It is a process of formation of sperm from a diploid cell. ഒരു diploid cell ഡിവിഷന് വിധേയമായിട്ട് ഒരു സെക്സ് സെല്ല് ഫോം ചെയ്യാണ്. ഒരു ഡിപ്ലോയിഡ് സെൽ ഡിവിഷന് വിധേയമായിട്ട് n നമ്പർ ക്രോമസോമുള്ള ഒരു പുതിയ സെൽ ഫോം ചെയ്യുകയാണെങ്കിൽ നമുക്ക് ഒരു പുതിയ സെക്സ് സെല്ല് ഫോം ചെയ്യുകയാണെങ്കിൽ നമുക്ക് അത്തരത്തിലുള്ള ഡിവിഷനെ എന്ത് പറയാം? സ്പെർമാറ്റോജെനസിസ്. During the time of spermatogenesis, the sperm may form from the cell having 2n number of chromosomes. What is the peculiarity of the sperm? Sperm may only contain half of the chromosome only. That is, it may only contain 23 chromosome in it. Understood, children? Do you have any doubt? No, isn't it? Before going to the topic of spermatogenesis, I'll tell you some interesting facts related to the spermatogenesis. Spermatogenesis will start when the male attains puberty. That is, when the male reaches the age of 10 to 16, their male spermatogenesis will start. Okay, that is during each cycle, during each cycle, male will produce millions of sperms, and these sperms are deposited in the vagina of the female reproductive tract during the time of coitus, and this sperm will survive inside the female reproductive tract till the 5th day of coitus. Okay, that means it will remain inside the female reproductive tract till the 5th day. This survival capacity of the sperm within the female reproductive tract is influenced by the quality of the cervical mucus. That is, cervical mucus quality ആണ് ശരിക്കും spermന്റെ സർവൈവലിനെ സ്വാധീനിക്കുന്ന ഒരു ഘടകം. Understood, children? Now can we move to the topic spermatogenesis? Okay, with your permission, I am going to start the topic spermatogenesis. Spermatogenesis involves a complicated sequence of events. This complicated sequence of events can be explained under 4 main phases. I'll write it in the board. Spermatogenesis involves 4 main phases. The first phase is multiplication phase. Multiplication phase. Okay. And the second phase of spermatogenesis is growth phase. And the third phase of spermatogenesis is maturation phase. And the fourth phase of spermatogenesis is known as differentiation phase. So now you might have understood what I have said. There are 4 main phases in the process of spermatogenesis. They are multiplication phase, growth phase, maturation phase, and differentiation phase. In multiplication phase, we will study the changes that may happen in the oogonia during the time of division. And in growth phase, we will study the changes that may happen in oogonia during the time of its growth. Okay. And the third, maturation phase, will explain about the changes that may happen in primary spermatocyte during the time of meiotic division. And the last phase is differentiation phase. In this differentiation phase, we will study the changes that may happen in the spermatid during the time of differentiation. Understood, children? These are the main things that we have to study. Now, let me explain with the help of a small picture. As I told you earlier, spermatogenesis involves 4 main phases: multiplication phase, growth phase, maturation phase, and last one is spermiogenesis. Spermiogenesis is otherwise known as differentiation phase. During the time of multiplication phase, the primary germ cells, which can be seen on the inner wall of the seminiferous tubule of the testis, will undergo mitotic division. As a result of this mitotic division, a large number of cells, spermatogonia cells, will form. These spermatogonia cells may contain 2 types of cells, that is type A and type B cells. These type A cells will act as a precursor of the spermatogonia again. That means it retains as a germ cell again, thereby it helps to maintain the number of germ cell. Okay, and type B cell will undergo some other changes. That means type B cell will enter the next phase of the spermatogenesis. In the next growth phase, the type B cells will undergo growth. As a result of the growth, it increases its size and acquire the shape of a primary spermatocyte. Now, the type B cell will acquire the shape of primary spermatocyte during the time of growth phase. Okay, and next the primary spermatocyte will enter into the third phase, that is maturation phase. During maturation phase, the primary spermatocytes will undergo meiotic division. That means the primary spermatocytes will undergo the first meiotic division. As a result of the first meiotic division, the primary spermatocytes will change into the form of haploid secondary spermatocyte. Here, due to the meiotic division, the chromosome number is reduced to half. Okay, I'll tell you once more. Here, during the maturation phase, the primary spermatocytes will undergo the first meiotic division. As a result of that, two equal sized haploid secondary spermatocytes will form. These haploid secondary spermatocytes will again undergo the second meiotic division. As a result of the second meiotic division, four haploid equal sized spermatids will form. So, the change of primary spermatocyte into the form of spermatid is known as maturation phase, or we can call the events. We can divide the events under maturation phase. Again, the spermatid, I told you, spermatid which is formed during the maturation phase will undergo some changes in order to acquire the form of sperm. So this is considered as the last phase of spermatogenesis. We can call this phase as spermiogenesis. That is, during spermiogenesis, the spermatid, circular shaped cell with all typical cell organelle, will change into the form of an elongated sperm through some changes. Okay, and these changes, or this change of a spermatid into the form of a sperm, is known as spermiogenesis. During the time of spermiogenesis, the sperm, major quantity of the cytoplasm which is there inside the spermatid will separate from the spermatid, and it form an acrosome at its tip and a flagellated tail at its end, and thereby it acquire the shape of a sperm. This process is known as what? Spermiogenesis. So after the formation of sperms, the fully matured sperm can be ejaculated, can be expelled from the seminiferous tubule to outside. The process of release of the sperm from the seminiferous tubule to outside is known as spermiation. This is the last step in the formation of sperm. That is, the release of sperm is the last phase of the development of sperm. Now you are watching the figure of human sperm. Human sperm is a microscopic long flagellated haploid sex cell which is produced inside the seminiferous tubule of male reproductive system. Its body consists of three main parts: a head region, a middle piece region, and a tail region. Head region contains a highly condensed chromatin body at its center. We can call this chromatin body as nucleus. The nucleus is enclosed within a membrane. This is the membrane. We can call this membrane as nuclear membrane. Just above the nucleus, you can see a cap-like structure. This cap-like structure is known as acrosome. Actually, this acrosome is made from the Golgi bodies of spermatid. And each acrosome contains certain hydrolytic enzymes in it. These hydrolytic enzymes helps to dissolve the membranes of the ova, and thereby it facilitates the process of fertilization. In addition to this, the head region, that is acrosomal region, contains some other polysaccharides also. Some specific molecules that can be seen inside the acrosomal region will help the sperm to recognize the ova of the corresponding species. Acrosin is an enzyme that can be seen inside the acrosome. It will be in an inactive state inside the acrosome. When the sperm head come in contact with the glycoprotein layer of ova, at that time this enzyme get activated due to the change in pH, and it will facilitate the sperm penetration into the body of ova. And here acrosome is occupied in a space which can be seen in between the plasma membrane and the inner nuclear membrane. That is, here we can see the acrosome. This is the plasma membrane and this is the nuclear membrane. Now we have to move to the second part of the body of the sperm, that is neck. Neck region can be seen in between the head and the middle piece region. And neck region contains centrioles in it. There are two types of centrioles in the neck region, proximal centriole and distal centriole. This proximal centriole will release certain elongated stiff fibers. We can call these stiff fibers as microtubules. These microtubules will facilitate the splitting up of cell during the time of cell division. And one more centriole will be there, that is distal centriole, that it can be found associated with the axial filament of the tail, and thereby it have a major role in controlling the lashing movement of the tail. The third part is the middle piece. Middle piece contain numerous mitochondria in it. This mitochondria will provide energy for sperm movement. In addition to that, it will facilitate the programmed cell death. Okay. And the last part of the body of the sperm is tail region. A tail region contains a plasma membrane in it outside and a axial filament inside. That is, tail contain an axial filament, and this axial filament is surrounded by a plasma membrane. The lashing movement of the tail will facilitate the movement of the sperm through female reproductive tract. I have said the parts of the sperm. I'll tell you it once more. The sperm contain three main parts: a head region, a middle piece region and a tail region. Head region contain a nucleus in it. Just above the nucleus, a cap-like structure can be seen, and this cap-like structure is known as acrosome. Acrosome contains certain hydrolytic enzymes and specific molecules for recognizing that ova, and in addition to that, it contains certain polysaccharides also. And the region which can be seen in between the head and middle piece is known as neck region. This region contains two centrioles, that is proximal centriole and distal centriole. Proximal centriole will facilitate the cleavage of zygote after fertilization, and distal centriole will facilitate the sperm movement also. And in the middle piece region, there are certain mitochondria. These mitochondria will provide energy for sperm movement, and in addition to this, it have a major role in programmed cell death also. Last part of the sperm body is tail. It contains an axial filament in it, and the axial filament is surrounded by a plasma membrane. You can see a plasma membrane here. Some certain adaptation which can be seen in sperm will help it to fertilize the egg easily. Here, the spindle-shaped body of the sperm enables the rapid movement of the sperm through the female reproductive tract, and the tightly packed mitochondria which can be seen in the middle piece provide sufficient energy for the propulsion of sperm through the female reproductive tract. Especially the molecule which can be seen inside the acrosome will help the sperm to recognize the ova of the corresponding species. Along with that, hydrolytic enzyme which can be seen inside the sperm will facilitate the easy penetration of the sperm through the membranes of the ova. Now, I think you are familiar with the structure of sperm. Today we have discussed about the structure of sperm and the steps of spermatogenesis. If you have any doubt, please contact me. I will help you. And stay safe, children.